Alright, today we're learning about flatworms, roundworms, and segmented worms. This is the worm lesson. A great question for me to ask on your final evaluation or on a test would be to compare worms. And if you have a good understanding of taxonomy and maybe phylogeny, you'll be able to combine those concepts and use that to answer the question very well. So as you work your way through this lesson, in your mind, try to make a table of comparisons and contrasts of these three worm phyla. The words themselves are quite a mouthful. We're going to start off with the Platyhelminthes and the nematodes. Remember that when I was doing the original order of the animals, and I showed you this diversity map here, that I gave you the basic definition of an animal. They all have more than one cell and some other things developed from a blastulus except for the sponges. And after that, we looked at symmetry. So every animal phyla has symmetry. And that gives us our cnidarians, which have radial symmetry. And after that, our animals develop bilateral symmetry, which I've talked about. Now, unfortunately, I'm not there to show you one of the videos, but one of the videos is going to talk about something called cephalization. So I'll try to talk about it in this flipped model here. So I did talk about the arthropods out of order because I wanted to give you an opportunity to work on your design an arthropod as early as possible. However, now I'm kind of going back to the order, and that's going to allow me to talk about the flatworms, Platyhelminthes, the roundworms, nematodes, and the segmented worms, which are over here. So as you can see, there are still a couple of phyla, the rotifers, which I'm not going to talk about, the mollusks, which I will talk about, and arthropods way here out of order. Okay? So let's go back to Platyhelminthes, and the evolutionary milestones that we're looking at are germ layers and body cavities for the two first worm phyla. So before I start talking about those particular phyla, let's look at the body plans that all bilaterally symmetrical animals have. When you go from radial symmetry to bilateral symmetry, what it means is that the animal now has a anterior portion where all the nerves, when many of the sensory organs are organized. So if I were to try to draw a diagram, I would have a radial organism that's kind of like a circle, I guess. It's a terrible drawing. But when we get this animal that has all the sensory organs on one end, it's now able to meet the environment from this end very well. So let's say we put eye spots to detect light, and we have maybe some kind of, or photoreceptor, maybe some kind of auditory oral receptor, ears of some sort. Maybe you have a mouth here that's going to detect taste and food. Maybe there's something else here that detects scents. But all these things are now in the anterior portion of the body. And so this organism is able to interact with the environment very well as it moves in this direction. So this can be a hunter, right? This over here, this radial symmetrical animal, it can do a very good job of interacting with organisms coming from the left, from the top, from the bottom, from the right. But bilateral symmetry does a really good job of interacting with organisms from one direction. And the word that we use to describe that is called cephalization, which is the development of a head. And cephalos is a Greek word for head. So you ever see cephalos, you know that you're dealing with the head or the head portion of the organism. So when you get to these bilaterally symmetrical animals, we have these body plans. And the body plans are named based on the fact that there is a body cavity. So the word that we use to describe a body cavity is the word coelomate. Coelom means body cavity. So now let's take a look at these prefixes here. We know that when we put the letter A in front of a word, it means the opposite. So asymmetrical versus symmetrical would be a good example. So body cavity, coelomate, acoelomate means no body cavity. And pseudo, we know from pseudopod and pseudonym means false, or false body cavity. So when I take a look at this graphic here, you'll see that I have the acoelomate body plan. It's a flat body plan. You'll notice there's an outer tissue layer, middle tissue layer, and an inside tissue layer, and there's absolutely no space. It's solid from the outside all the way to the in. And right in the middle, where there's that hollow space, is the digestive cavity, the intestines, stomach, all that kind of stuff. In a pseudocoelomate, you'll notice that there is an outside layer, there's a middle layer, the pink, and then there is some space. And then we get to the inside layer. And that space between the pink, middle, and the yellow, inner, that is the body cavity. So as you can imagine, this organism rolling around, like picture that being you. As you went on your back, your belly would flop to your back. And when you went on your front, your belly would flop to your front. So this organism here, their body cavity, is not a true body cavity because their organs inside can just flop around. Which, for a small organism, not a big deal. For a large one, like us, it just wouldn't work out very well. And then we get to the highest evolutionary concept of body cavity, whereas a true body cavity. So taking a look at this, you can see that we have the outer layer here, we have the middle layer, and then we have the inner layer. But notice the inner layer here is surrounded by tissue and is held in place by this thing right here. And this tissue right here is known as mesentery. So the purpose of the mesentery is to hold the inner tissue in one spot. So this organism goes to the left or the right, its organs won't flop all over the place. So this allows an organism to grow larger and still be effective. If you're small, you don't need a body cavity. You can rely on diffusion. If you're another strategy is this pseudocoelomate, but if you want to get large and be super efficient, then you do need a true body cavity. Now you'll notice that I'm using this terminology: outside layer, middle layer, and inside layer. There's actually names for these. These are the germ layers. So when an organism develops in embryo, which we looked at in the evolution unit when you guys did comparative embryology or evolution, evo-devo, these germ layers, they start very, very early. The zygote, it splits under mitosis and it goes into 4 cells, 8 cells, and eventually these tissue layers develop. And so I'll talk more about the blastula development later on when I deal with echinoderms, but for now, know that animals have 3 layers. They have an inside layer, the yellow, which is known as the endoderm, right? So the endoderm is yellow. They have a middle layer called the mesoderm, meso for middle, and they have an outer layer, ectoderm, ecto meaning outside. So those prefixes again: ecto out, meso middle, endo inside, derm is skin layer. And so each of these germ layers, they all develop into very specific parts of the body. Okay? So now that we understand, there's a lot of terminology. These are evolutionary milestones. So one of the evolutionary milestones is the development of a body cavity. So going from no body cavity, acoelomate, to a false body cavity, to a true body cavity. And also the presence of tissue layers. That's another evolutionary milestone. So no tissue that you see in the sponges, to 2 tissue layers that you see in the cnidarians, to 3 tissue layers that you see in everybody else. So everybody else, all the other phyla, have 3 tissue layers. Okay? So that allows me to go to the very next phyla. We've done sponges, we've done cnidarians, and now we're doing Platyhelminthes. So your Platyhelminthes, there's the word there, they are represented by 3 classes. And the 3 classes, you might recognize them as tapeworms. The proper name is Cestatoa. Planarians, which we're going to see in the classroom on Monday, and the proper name is Turbellaria, and flukes. And if you've got a cat or a dog, you do have to be concerned about this because they are often infected with flukes. And if you don't cook your pigs properly, you can often get flukes as well. And the proper name are trematodes. So these are 3 classes. So remember that when you're creating your map, your tree. Your defining characteristics of your Platyhelminthes are on this slide. So from now on, every single organism that we're going to talk about has bilateral symmetry at some point in their life cycle. So that's true. And they all have 3 tissue layers. That's also true. So that's a characteristic that you'll see for Platyhelminthes and everybody else from now on. They also have a centralized nervous system. Remember our sea anemone and how it just, all of a sudden started swimming? It was really amazing because it doesn't have a brain and it can swim away. But the Platyhelminthes, they do have bilateral symmetry. They do have a head. So they have a centralized nervous system that allows them to interact with the environment from one direction. They have those 3 tissue layers, the endo, meso, and ectoderm. They do not have a body cavity, so we call them acoelomate. And because they don't have a body cavity, they don't require any systems to move nutrients and oxygen around. They can just rely on diffusion. Okay, so that's the characteristics of your Platyhelminthes. So looking at some of these pictures, the Platyhelminthes has a really weird mouth. We're used to the mouth being inside the head, but their mouth, or pharynx, their throat, is actually halfway down their body. And they're able to stick that into whatever they're eating and just liquefy whatever they're eating and suck it into their body. There's their nerve net that goes all around their body, and it's centralized on their anterior region or their head. They're carnivores. They eat other organisms. They eat meat. And when we bring the planaria in, we feed it liver. So we get liver from sobies, cut it up, it's a little bloody. They like that stuff. Your flatworms, they can live in hosts as a parasite, or they can live free in the soil or in the water, in fresh or salt. And in terms of feeding, well, they're most, notoriously known for their parasitic relationship. You'll see things like Grey's Anatomy, which many of you seem to watch, and there'll be an episode where this girl has decided that she wants to go on a diet, and so she ordered a tapeworm from some mail order place, and it's down in her belly, and she can eat all the food she wants, and she doesn't gain any weight. However, she has a worm inside her body that's 30 feet long, and that's just not healthy. Anyway, that's more science fiction than reality. When people get these parasites, they're not trying to get it on purpose. It's often because of uncooked food or going in a bad location. So remember, you're responsible for knowing the life cycle for one representative life cycle for each kingdom, and for the animal kingdom, you have many choices. I've given you some for the other two. There's also a life cycle here for the blood fluke. In grade 10, some of you did the climate change and disease, and one of the diseases you did was schistosomiasis, and it's a disease that is carried by the snail as a primary host, and unfortunately, if you go into the wrong area with a bare foot into a pond, or the stagnant pond, or dirty pond, then the fluke can leave the snail, swim in the water, and burrow a hole into your foot, go into your bloodstream, go into your digestive tract, and eventually reproduce itself, producing tons of eggs. In your digestive tract, you then poo out. You do it in the toilet, but if you're another mammal, you poo it out, and then the snail would come along, pick it up, and the cycle continues. So that's one life cycle. Another life cycle is the tapeworm. So for those of you with cats and dogs, you often need to make sure you get them sprayed and they get their shots properly so that they don't collect tapeworms. Tapeworms can occasionally be transmitted from the pet to the human. It's not very often, because most of you do not go to the cat litter box or the dog poo and pick it up with your hands and then start eating your food without washing your hands, so it's not really an issue, but it can harm your pets. Fleas are typically a vector that will carry the tapeworm, and when they bite the cat or dog, they'll inject the tapeworm into its body, and it'll grow. In the video, which I would like to show you, I would have shown you a tapeworm that lives inside the human host. So they live inside our stomach, and it's not common, but if it does happen, it's quite unpleasant. I do have a couple of sites here which you guys can look at, and in terms of the flatworm, I'm going to put a link that I would like you to watch. So you can't watch the full video, but you can watch this 10-minute video, and it gives you some information about flatworms, and it'll allow you to fill in the collaborative document that I've put up online for you guys to do as a class. So it gives you a little bit of information here about the flatworm, and so you'll see a rich diversity. They're actually really remarkable animals. And in our flatworm lab on Monday, you're going to cut, whoever chooses to do it, you're going to cut some flatworms in pieces and see which parts regrow. And once you watch that video, you can click on this link here that says About Flatworms, and you'll get to this page here, which gives you lots of information about flatworms. This video, I haven't watched it, but it says it talks about the evolution from bilateral to, from radial to bilateral, so that might be a good one to watch. And there's some other videos here about diversity and diseases and the parasites and the free living forms. So if you want to get a little bit more information, this would be a good page to go to. Now, with each of the phyla, I'm going to talk about some kind of human impact, whether or not it's climate change impacting them or how humans have done something around them. And with the flatworms, if you go back to my PowerPoint here, you'll see that here's my STSE connection. So there's going to be a question on your test, and it's going to be some kind of human impact question. And you can answer. For level 4, you're going to want to have several examples from various phyla of how humans have impacted the biodiversity of the world. And you can use any of the examples I give you. I'll probably give you 10 or 15 examples, and they're written down. I'll talk about them. There's videos and so on. So you can read this clip. And basically what happened was there was a bunch of farmers in England, in the United Kingdom, and they were noticing that their crops weren't doing as well. And eventually their crops were dying, and they couldn't figure it out. And then they realized that the earthworm population was being decimated. And so these were earthworms that were brought over from Australia or New Zealand. And these flatworms, what they were doing is they were eating the earthworms. They were destroying them. They were just eating hundreds a day. And so they were completely decimating the earthworms, which are necessary for a healthy ecosystem. So here's that hammerhead flatworm. There's some really interesting videos online. You can look at them and see them. But we need the earthworms in order to turn the soil and make sure it's healthy. But without them, then you can't grow things very well. So there's lots of videos on this. I'll put some links up on the website so you can take a look at them. People in the UK are inspecting their plants and their pots to make sure they don't carry any of them into their own property. But it is it's a losing battle right now. They're losing that battle with these giant predatory flatworms that eat our earthworms. There's a picture of a flatworm here attacking an earthworm, and it will basically liquefy it and then suck it into its body and move on. And so they're eating them like crazy. So there's a human impact connection. We brought a species over from potted plants from another country, another continent, to a place where there's no natural predators, and they've reproduced like crazy, and they're decimating the local ecosystem. Okay, so that's the end of my flatworms. I'm going to move to roundworms next in the next video.